Our next speaker is Professor Alistair Boxall. He's no stranger to Ireland. He has a grow for Donegal, I understand. So he's going to speak to us about the emerging issue of AMR, or the issue of AMR that we thought had gone away. So over to you, Alistair.
Thanks, Maurice. So it's great to be here. So I am going to talk about AMR, but I'm going to talk actually about pharmaceuticals and the environment more generally. So I'm going to talk about things that we use for our health and discuss how they could be impacting the health of the environment, but also how that can then come back and impact our health. So it's a bit circular, really. So what I'm going to do in the next 17 minutes, I think I've been given, is tell you a little bit about pharmaceuticals in the environment and to talk about impacts on ecosystems and also we'll focus a little bit on antibiotics and antimicrobial resistance. So I come from the UK, so I'm based in York in the UK. And in the UK, if you go to the National Health Service website, you can download all of the prescription information that we prescribe each year in the UK. And we use about 1,200 different active pharmaceutical ingredients. In terms of tonnage, in terms of prescriptions, we use about 13,500 tonnes of pharmaceuticals in the UK each year. So we're talking large numbers of molecules, and actually, I think compared to the PFOS, a little bit lower, but I think if we times it by 30, not that far off. So fairly large quantities of these things. And about 10 years ago, one of my PhD students did a survey to try and find out what happened to the pharmaceuticals that we use. So she went out into York, she spoke to people in the city, asked them, what prescription medicines do you use, what over-the-counter medicines do you buy, and what happens to those. And we have the data just summarised here. So for the prescription medicines, the good news is the bulk of those are used, which I guess makes sense. When we go to the doctor, we'll be prescribed these things and we will use them, but some of those won't be used. The amount of use for over-the-counter medicines is a little bit lower, but we do have fairly large chunks of medicines that aren't used. Some of those are taken back to the pharmacist, so if you have unused medicines that you want to get rid of, certainly in the UK, that's the way to dispose of it. It will go back to the pharmacist, and the pharmacist will then dispose of it as hazardous waste. But some people, particularly things like cough medicines, will pour those medicines down the toilet, or they'll throw the unused drugs in the bin. So they'll either end up in a landfill site, or in the UK, certainly around us now, we're moving more to waste-to-energy plants, so those nowadays will end up in incinerators. But what we can do with that data is we can do a bit of a back-of-the-envelope calculation. So of our 13,500 tonnes of active pharmaceuticals, about 2,800 tonnes of those will be unused, and they'll be then going down those different routes, so thrown down the toilet, thrown in the trash, or back into the pharmacist. But of the medicines we use, this is for the prescription medicines, just under 5,000 tonnes will be metabolised, but what that leaves is about 6,000 tonnes that will be emitted down the wastewater system. So they'll enter something like this. So this is, I think, a really nice picture of one of the Victorian sewers in London. So you can see the wastewater coming down through the sewer system. And if I took a sample of that water and I analysed that, I would find pharmaceuticals that we use as society. I'd also find other chemicals that I use as society. Now that wastewater then will be transported to some form of treatment works. So in the UK, and I think it's very similar over here, we have two types of wastewater treatment works. One is a very active process. It's called an activated sludge treatment system, where you have almost like something the size of a swimming pool. You're bubbling air into it, and the idea is that the bacteria will degrade the sewage. This is the other type. So this is a trickling filter. What you have here is a bed of stones, and on those stones you have a biofilm. And that biofilm is degrading the wastewater, so it's degrading the organic material in the wastewater. Now the problem is these were never designed to treat out things like pharmaceuticals. So while they're quite good at removing some pharmaceuticals, so something like paracetamol which is really heavily used in the UK, is actually quite well removed by these things. Other pharmaceuticals will basically pass straight through the sewage treatment system and they'll end up in your local river. So this is one of the rivers that we do a lot of work on. So this is the River Foss, which is quite close to where I live. Just upstream of this site, there's a wastewater treatment works. It's getting wastewater from about 20,000 people. And actually, this is the most polluted spot in York in terms of levels of pharmaceuticals. So that link then between pharmaceutical use and the environment was made just over 25 years ago. So it really kicked off in the late 1990s. In 2002, the study at the top came out. So this was a study done by the US Geological Survey, where they went out across the US to 139 different locations. These were rivers and streams and lakes. They took samples of water and they analysed that water for a range of emerging contaminants, including pharmaceuticals. And the conclusion from that study was that pretty much across the US, pharmaceutical contamination of rivers was pretty much ubiquitous. So they were detecting pharmaceuticals in about 90% of the samples that they analysed. That paper's had massive impact, so I think it's now the most highly cited paper in the journal Environmental Science and Technology ever. So it's really kicked off a whole area of research. So these types of studies have now been done all over the world. This study came out in 2016. It was funded by the German Environment Agency. And what they tried to do is look at what we know about pharmaceutical pollution around the globe. So what this map here shows is basically what we know about the occurrence of pharmaceuticals. So the darker the red, the more we know. So countries in Europe, countries in the US, we have data probably on a few hundred different pharmaceuticals in surface waters. But as you move down to the yellows, so we've got some yellow countries here, there's only data on 1 to 3 pharmaceuticals, and that's probably only at a couple of sampling points. And then you can see that we've got quite big areas of grey, and those areas we know nothing at all, or then we knew nothing at all about the level of contamination from pharmaceuticals. So about 5 years ago, we decided to fix that. So we kicked off what we've called the Global Pharmaceutical Monitoring Project. We've worked with about 100 collaborators from around the globe. And what we've done is we've worked with those collaborators to get samples from rivers all across the world. So we've covered 104 countries. That's involved 134 sampling campaigns, over 200 different river systems, and 1,050 sampling locations. And basically what we did is we shipped these kits out to the collaborators. So you can see this picture up here. There's a box. So these were the boxes that we shipped. These could fit in the palm of my hand. They were shipped out to the collaborators. In that box, you had sampling vials. You can see we have a little bucket and a string so they can take the sample. We have syringes. We have filters so they can filter the sample. And the idea would be that they would then go out to their local river. They'd go to different points along their river, take a sample. They would then freeze that sample, ship it back to York. Usually they get back to York in a day or two using DHL, and then we would analyse them for 61 different pharmaceuticals to get an idea of the level of contamination. So here's just a picture of one of those samples being taken. So this is actually one of the lowest flow systems that we looked at. This is actually the Alexander River in Israel. It's a really interesting river, and we might pick up on it in the discussion later. But basically it originates in Palestine, comes over the border into Israel, then flows through Israel and then out into the Mediterranean Sea. And that river then is being used for irrigating crops. So it's being used as a source of water. You can see in this particular time when we sampled it, it's very low flow. So obviously Israel is a very low flow system. And basically what you get in that system is you've got raw sewage coming into that river, very little dilution, so we see quite high concentrations of pharmaceuticals. This is a site in Nairobi. So this is Lydia, who was our collaborator in Nairobi. And you can see here we've got a picture of Lydia taking a sample of one of the sites in Nairobi. So this was a river, or a tributary of the Nairobi River that went through the university campus. So as I say, those samples then came back to York, where we analysed them for 61 different pharmaceuticals. I could spend the whole afternoon talking you through the data. I think there's 120,000 different data points, but I'll try and summarize it in just a couple of graphs. And the one that I'm going to show first is the average overall level of pharmaceutical pollution at the top 26 most contaminated sites. So it says 25 there, but it's actually 26 because I wanted to include Scotland, which is over there on the far right. But you can see the most contaminated site was Lahore in Pakistan, and that was followed by Bolivia, and that was La Paz in Bolivia. So those were the most contaminated systems. The most contaminated location was actually in La Paz in Bolivia, and that's a photo of that site. And basically, if you go to Google Maps and you look at that site and you look at the surrounding area of that site, you find there's quite a lot of pharmaceutical manufacturing plants situated around the river. So we think what's happening is, you can see it's quite a low flow river, but you've probably got waste from that pharmaceutical manufacturing area coming into the river. Again, it's not being very heavily diluted, so you're seeing quite high concentrations of pharmaceuticals. Generally, we found the most contaminated sites to be in Asia, Latin America, and Africa. But you can see that Europe does figure. So we have Spain is here. So this is Madrid. And then you can see we have the UK over here. So that's the River Clyde in Scotland. In terms of the pharmaceuticals we detected, of the 61 that we monitored, we detected 52 different active ingredients. The most common ones were paracetamol. So paracetamol was the highest concentration pharmaceutical that we detected. So that I think just reflects the fact that pharmaceuticals are available everywhere, very widely used. Number two, and I doubt whether this is due to pharmaceutical use, although it is used as a pharmaceutical, is caffeine. So caffeine came in at number two. And then number three is metformin. Now metformin is a treatment for type 2 diabetes. And this is a molecule that we find pretty much everywhere at very, very high concentrations compared to other pharmaceuticals that we looked at. In terms of how widely we detected these things, what we have here is an assessment of the frequency of detection. So the most frequently detected drug was carbamazepine. So carbamazepine is a treatment for epilepsy, and we saw carbamazepine in over 60% of the locations. This is 1,052 locations that we monitored. Number two, again, is metformin, the type 2 diabetes treatment, and then we have caffeine coming in at number three. Over here, we have an assessment of the mixtures. So we just heard about cocktail effects. What we're able to do with each of the samples is basically get an assessment of the number of chemicals that we detect in each sample. And that's basically shown in this graph. So we have the number of compounds in the sample here, and then the frequency or the number of samples where we saw that number of compounds. So if we take this bar here, basically what that's saying is 100 of the 1,050 locations we detected only one pharmaceutical. But if you move over here, we see one site, so one water sample, where we detected a mixture of 34 different pharmaceuticals. And I'll come back to that later. So I think what this is showing is that pharmaceuticals are getting into the environment. They are pretty much everywhere. And that then raises the question, well, so what? Now, the problem is these molecules are designed to be biologically active. So in you and I, they're designed to interact with a receptor or a biochemical pathway. And the problem is a lot of the receptors and biochemical pathways that are in us also occur in fish and also occur in invertebrates. So the concern is that if these molecules are getting out into the environment and they're accumulated by a fish, then they're likely to have some effect on the fish at very, very low concentrations in some instances. This is just an analysis that was done by the University of Exeter. This is where they took DNA information to look at the presence of receptors in organisms in the environment, and they estimated that about 90% of the receptors, the pharmaceuticals that we use that are targeted by pharmaceuticals actually occur in organisms in the environment. So it's over 90%. So with our data then, what we've done is we've taken, or we've used the sort of published data on ecotoxicity of these molecules to back out what we think are safe concentrations of molecules in the environment. And then we compared the concentrations that we see at each of our sites with these safe levels. Now the headline is that 43.5% of our 1,050 sampling locations contain at least one pharmaceutical at a concentration above the safe level. And if we go back to the comment about cocktail effects, that's just considering single compound effects. It's not considering the potential combination effects of these molecules. So you could get these working in synergy and actually enhancing the effect in the environment. So I think that's a fairly significant chunk of the samples that we see that we think are at risk. But just building on that mixture thing, it's not just an organism being exposed to a mixture of chemicals, it's mixtures of organisms being exposed to mixtures of chemicals. So if we go back to that site in Hong Kong, this was the site where we saw 34 different drugs, and we look at the information we have on the effects of those drugs, what we find is that some of those pharmaceuticals will be affecting the growth of the algae in that system. That algae is obviously being used as a food for the invertebrates, so you're affecting the food for the invertebrates. But you've also got chemicals in that mixture affecting the invertebrates themselves. So you've got the combination of the effect on the food and the direct toxic effect on the organism. Those invertebrates are going to be then fed on by the fish, so you're affecting the fish food, but the chemicals will also be affecting the biochemistry of the fish, they'll be affecting the histology of the fish, and they'll be affecting the fish behavior. Things like antidepressants will be affecting how alert the fish are and possibly making them more prone to predation. So when we consider these mixtures of chemicals and mixtures of species, we've got all of these complex interactions happening which could really exacerbate the impact on the environment. And I think this is something that we're very keen to look at in the future. There's also human health impacts. So we looked at 20 different antimicrobials in the study, and we detected, I think it's 13 of those antimicrobials. And obviously, I think we're going to hear a bit more about antimicrobial resistance later on, but there's big concern now about antimicrobial resistance and the antimicrobial resistance crisis. At the moment, it's killing millions of people a year, but in the next few decades, that's forecast to increase to tens of millions of people a year. And we're beginning to recognize that the environment could well be playing a role. So this really kicked off in 2007. This study by Joachim Larsson's group in Sweden was published, showing very, very high concentrations of antibiotics in areas of India where you had drug manufacturing. And what they showed is that levels that we're seeing in water were actually equivalent to the levels that you see in the blood of someone who'd been treated with some of these antibiotics. So these are scary levels. So what we can do with our data is begin to take our data and compare the data to levels that we think are safe in terms of resistance selection. And our calculations indicate that about 17% of our 1,050 locations have concentrations of antibiotics that we think are a concern in terms of resistance selection. You can see also that those sites are concentrated you've got Bolivia there again, but a lot of them are in Africa. So we've got countries in Africa where almost every sample that we see, or we took, had levels of pharmaceutical antibiotics above levels where we think they could be selecting for antimicrobial resistance. And that could be really important in terms of the AMR crisis. This is the worst site in terms of antibiotic pollution. So this is actually a site in Bangladesh. You can see our collaborator there taking a sample. You can see some houses in the background. And again, if you go to Google and go to Google Maps and look at the area behind that, again, it's drug manufacturing. So it's a molecule called metronidazole that we were detecting in that water, and we suspect that the factories are probably formulating or manufacturing metronidazole when we were sampling that site. So hopefully that gives you an overview for what we've been seeing in terms of pharmaceutical pollution around the globe and what the implications are. I think hopefully I've convinced you that these molecules are being released to the environment. They're being released at all stages in their life cycle, from production right through to use. In terms of contamination, we see the biggest problems in low and middle income countries, so areas of Latin America, Asia, Africa, and we see a real mix of different pharmaceuticals in those waters. And what we're beginning to find is that a significant proportion of those sites have levels that exceed levels that we think are safe in terms of ecosystems and also human health. So I think this is a thing that we really need to start trying to fix, and we need to begin to get these levels down and so clean up rivers globally. I think what we've also shown is the power of global collaboration. So we delivered this study probably for about 50,000 euros in terms of cash. It's obviously involved a lot of staff time, but just the collaboration, people offering their free time to go out and sample. The analytical methods that we have available now, they allow us to do this stuff really quickly. And I think that's very powerful. And what we should be doing is extending this type of work to a much wider range of environmental contaminants. So I'll shut up there. That's my email, in case anyone wants copies of the papers. So there's 3 papers that sit behind the talk. Happy to send those if you want a copy. We also have a website for the project, which is www.globalfarms.org. Thank you.